sort of future scope a little bit some really important ideas in terms of ocean global change, and they tried to sort of get us to talk about some of the work we were doing in some of the areas we thought were really important. So I'm going to talk about that today. And primarily, the work I've really been focusing on the last few years is evolutionary potential to climate change, and that's what I think is what interests me at the moment. So we all know the ocean is changing, the physical parameters of the ocean are changing, it's warming, and we're seeing ocean acidification as a result of increasing CO2 in the atmosphere. But primarily what we really want to know is how will marine species respond? What are the organismal sort of responses that we're going to see, the community responses that we're going to see? And the way we often do this is that we conduct some sort of manipulative experiment where we might bring animals into the lab or rear them in the lab under some sort of combination, let's say warming or acidification, and we might rear them for some relatively short period of time, usually in the order of hours, days, weeks, or maybe months if we've got a really what we consider a long-term experiment, and we look at how they perform under those sort of predicted future conditions, and we then use that to extrapolate what might happen under climate change. So we're using these sort of short-term experimental manipulations of future climate change conditions to predict what's going to happen 50 or more years out into the future. Now, of course, there's some real limitations to that because these sorts of experiments actually really risk overestimating the impacts of climate change. They could go the other way, but potentially we're worried about overestimating here. And just to give you a really simple example, if we were to bring 100 fish into the lab, we stick them under warmer conditions, and we find that half of them die, as ecologists we might sort of wring our hands together and say, oh, this is all very terrible, half of them died, well, the population is going to go extinct in no time if it gets warmer. An evolutionary biologist might look at that and say, wow, that's amazing, half of them survived. Actually, maybe there's some genetic variants in there that might allow them to adapt over a long time frame. So we need to have sort of a realistic sort of expectation or understanding of what we might be getting out of short-term experiments and consider the longer-term impacts. And of course, there are two main ways that animals can respond to climate variation over the longer term, adaptive responses that they can make. And that's either through acclimation, and this is plasticity, and this is physiological, behavioural, or morphological adjustments without genetic selection. So it's what the actual genotype can do, plasticity in the genotype to respond to environmental change. Or we can actually see genetic adaptation, and that's selection on genetic variation that's inherited from one generation to the next, such that we see a shift in the allele frequency of the population through time. But basically, we might be expecting to see the same thing with either of these responses, that sometime in the future, the population would be better able to cope with a warmer environment than it can right in the current day. Now, plasticity is thought to be a particularly important way that animals can respond to climate change because it can be fairly rapid. So you can see rapid phenotypic responses to climate change, to environmental change. And it can enable performance to be maintained in a new environment, so it can be beneficial. And it can potentially allow adaptation to catch up over the longer term. So it can sort of buffer change in the phenotype in the population and allow adaptation to catch up over the longer run. And one particular type of plasticity we're really interested in usually in climate research is a thing called phenotypic buffering, and that's where the observed functional phenotype that we're interested in pretty much stays unchanged with some sort of change in the environment. But there's plasticity in a whole bunch of other things that underlie that. These might be gene expression or behavioural or morphological changes in energy allocation, those sorts of things. So there's plasticity there, but the functional phenotype is being maintained. And we might also think about different types of plasticity when it occurs in the lifetime of the organism. The one that most people really think about is reversible plasticity. This is things that, for example, happen in adults, might allow them to acclimate to seasonal cycles. So they adjust their metabolism, for example, for winter and summer, or even daily cycles for things in tide pools and things like that. But there are other ways that plasticity can occur, there can be developmental plasticity, and this is when an animal sees a particular environment very early in life, and that sets in train a series of developmental processes that leads to an altered phenotype that might actually then help them in that environment later in life. And developmental acclimation or plasticity is actually quite common. Or we can get transgenerational plasticity, and that's where the environment that was seen by the parents or even the grandparents influences the offspring in a beneficial way, usually. So there's sort of it can be short-term or quite long-term, and different conditions would actually promote different types of plasticity. Okay, so we're interested in coral reef fish. That's what I do in my lab, and we've been rearing fish under higher temperatures in particular for quite a length of time. And what we see in general is that if you expose them to somewhere between 1.5 and 3 degrees higher than the average that they see in summer, that you can see reduced performance in things such as growth, reproduction, swimming ability, aerobic performance. So there's a range of declines in the fitness of the animal. And it's not just in things like the little spiny damselfish that we rear, but also other studies, work done here by Morgan and collaborators have shown sort of similar things in coral trout and some other fish. And the other thing that we see is that generally there's limited capacity for this reversible acclimation. So it doesn't really matter how long we hold these animals under higher temperature, they don't improve their performance. So they haven't got much scope, it was seen, for reversible acclimation. And that's been seen by a few other people as well. So what we've been doing here is trying to rear the fish over multiple generations to look for potential for developmental and transgenerational acclimation in particular. So will these populations potentially improve their performance over a couple of generations or under much longer term rearing? Now, this is work that Jenny Donaldson really pioneered nearly a decade ago now. And really this has been a fantastic experiment she really pioneered and deserves an awful lot of credit for. And I don't think even Jenny realised when she started this just how much this would continue to give us a fantastic experiment. And so what Jenny did here was she brought in breeding pairs of the spiny damselfish as our sort of lab rat and kept them at current day temperatures, so they're all actually adjusting to a common environment, get rid of some of the parental effects that might have been out there from wild caught animals. Then she bred them, the F1s, then they go into the current day conditions again, or warmer temperatures, plus 1.5 and plus 3, then they go through to the F2s and they're split again, except that in the higher temperature lines they're just continued through. And actually this runs through to the F3s, and Jenny's in the process of actually sort of dissecting that data at the moment. But I'm only going to talk about up to the F2s at the moment. It's complicated enough up to the F2s without worrying about the F3s. And then you test the fish at current day summer conditions or the warmer conditions, plus 1.5 plus 3. And this is a really powerful design because it allows you to do three things. It allows you to look at the acute effects of high temperature, what sort of these short-term experiments tell you. I'm just sort of plonking them in warmer water. Developmental effects, developmental plasticity can be teased out here, because here you've got some fish that are current day and others that have been reared up since early life under higher temperatures, and transgenerational effects, because here you've got ones that are reared up under higher temperatures, but their parents were under higher temperatures as well. So it's actually a relatively simple but very powerful design. And this is one of the first lots of results that we saw coming out of this experiment. It was really quite exciting. So this is aerobic scope. You don't need to worry about that too much, other than it's an indicator of the capacity to do aerobic activity in the fish. It's the difference between resting metabolic rate and maximum metabolic rate. And so it might tell us something about the potential fitness of the animal. So we have aerobic scope. This is the acute treatment. This is the aerobic scope they achieve under control conditions, the normal temperatures that they experience. And if you put them in acutely into high temperature, their aerobic scope plummets dramatically, and we've seen that before. So it's nice to be able to repeat that. This is what happens under developmental Acclimation, so this is the potential for developmental plasticity. And what we see is there's some improvement here, particularly in this group here, where they've had +3 all their life since hatching, then aerobic scope goes up. So it does seem there's some developmental plasticity there. They do a little bit better, but not really in the +1.5 group. Where things got really exciting was when the parents had been exposed to higher temperatures. And here we see complete restoration of aerobic scope, and if anything, probably an overshoot of aerobic scope. So they do just as well, if anything, they do better than fish that were in the main control group. And so clearly the parental environment has a massive effect on how you would assess whether these fish are susceptible to climate change or not. So that was pretty good news. And since that time, there's been quite a few other studies that have shown very similar things. And there's now a range of studies showing parental effects or transgenerational effects on temperature and growth in things such as the sheep's head minnow and stickleback from temperate waters, high CO2 and survival in the silverside, and high CO2 and growth, which is work that Gabby Miller did here in the cinnamon clownfish. So this seems to be something that we really need to take notice of. It's not just some funky thing that's strange with the spiny damselfish. What we've also been able to do more recently is really value add to this and sort of engage with some people that do really good molecular work from the Kaus group and with people here, with Heritability, for example, and start to look at some of the mechanisms that underpin this. So we've started working a few years ago, actually probably about 6 years ago now, with Tim Ravassi's group at Kaus, and we've been able to look at what the actual mechanisms are of this transgenerational plasticity we see. And it's really interesting that by looking at the transcriptomes, the whole transcriptomes of the animal, all the genes that are being expressed under these different treatments, we can see that there are changes in metabolism, which suggests that there's shifts in energy production that allow for this transgenerational plasticity. There's also changes in things like immunity and stress, tissue development and transcription regulation. So we can start to look at the actual processes that are underlying this. And even more importantly and interestingly, we can actually try to see how this happens, not just which genes are involved, but how do you get this transmission of plasticity across generations. And it looks like it might potentially have an epigenetic basis. So we've now done whole genome transcriptome work, which is sort of really cutting edge epigenetic work. I should have been in the main transcriptome, I meant methylation. So this is really cutting edge epigenetic work that suggests that there's differential methylation going on in the transgenerational groups. And we've actually identified 193 differentially methylated and aerobic scope correlated regions. So these are regions that are differentially methylated in the genome and they correlate with the phenotype. And so and really interestingly, those regions, some of the genes that are involved in those regions are related to metabolic homeostasis, insulin sensitivity, and insulin is really important in energy production and oxygen delivery. So all the sorts of processes that we might think would be associated with change in aerobic scope. And so this is actually gives us some confidence that we might actually be looking in the right place here at what's going on. And so this is just to illustrate that we can know from a relatively straightforward experiment, we can actually add some very cool molecular work and understanding to some of this. Now, I don't want to sort of stand up here and suggest that, well, this is all fantastic, just means you've just got to expose the parents to warmer conditions and everything's going to be all right, no? We can just walk away, climate change, you don't have to worry about it. And that would nearly be what you'd expect if you just saw that. But it turns out there were some limitations. So aerobic scope was restored, but when we got to the F2 and when we got those fish to reproductive age, they didn't reproduce. And that's about as bad for fitness as you can see. So this is all very good. Aerobic scope looks great, but reproduction doesn't look too good. So suddenly it didn't look so good after all. But luckily, Jenny was even smarter than I thought. She did something else. In this experiment, not only did she have these lines that went straight through, but she had this really interesting treatment where she took the fish that had been reared up under 1.5 in their first generation, under as F1s, and then rather than just taking them through, she bumped them up another 1.5. So we had this stepwise increase in temperature across generations. That's probably much more sort of realistic or representative of what's happening in climate change, rather than just throwing these animals into plus 1.5 or plus 3 for a couple of years and seeing how it goes. And so we had this stepwise treatment. And the really cool thing about that was that even though reproduction was depressed in terms of clutch size and number of eggs, 70% of the pairs bred when they were raised 1.5 per generation. And so this suggests that actually how we do these experiments actually matters, and that a really sort of that real press that we had in the first generation may not have been the best thing to do, and that these stepwise increases might be really important. The other thing that we're able to do here is also use this molecular approach to try and see what was the difference between the stepwise and the transgenerational treatment. Were the genes in common? Were the genes that were different? That might tell us something about what processes allowed the aerobic scope to be restored, but not reproduction, only reproduction to be restored in the group that went through the stepwise treatment. And we did in fact find 32 differentially expressed genes that were in common between the step and the transgenerational group, and they tended to be related to metabolism and respiration, those things that might link to aerobic scope. And there was also 123 differentially expressed genes that were different between the transgenerational and the stepwise group. And these were things that are involved in mitochondrial function, inflammation, and DNA repair. And maybe they have something to do with their ability to reproduce in those conditions. Just illustrating again that some of these techniques can actually be quite informative in terms of telling us a little bit about the mechanism. Okay, so that's a little bit about plasticity. What I'd like to move on to now is talk a little bit about genetic adaptation or evolution. And remember, this is selection on genetic variation that's inherited from one generation to the next. And I'm going to show you this again. Because it turns out that this was actually really, really useful for looking at a question of adaptation, because we knew where every individual went. So Jenny kept track of every single fish in that experiment, and so we knew everyone's mum and dad, everyone's grandparents. So we had an entire pedigree that we could actually look at genetic variation and estimate heritability. Now, for those of you that have forgotten your undergraduate evolutionary biology and haven't opened an evolutionary biology book ever since, I'll just remind you that heritability is notated by H squared, and it's the proportion of phenotypic variation that's attributed to additive genetic variation. So we're looking for heritability. We can do this in this sort of design, and we use a thing called the animal model. It's a fancy statistical method to actually tease out the proportion of variation that can be attributed to additive genetic variation. And when we do that, we find that the traits that we've been looking at, the metabolic traits, are highly heritable. So the phenotypic variation has a heritable component. And importantly, it's not just at the sort of current day temperatures, but also at the higher temperatures, which are notated here, 0, 1 and 2. That's the current day, 1.5 plus 3. And all of them have very high heritability. Heritability goes between 0 and 1, and so these are really quite high heritability, up to 0.78, which is very high. So the traits are heritable. So that's cool. They're heritable. They're heritable at high temperatures. They should evolve. But even more important is to look for gene by environment interactions. So not just whether they're heritable, but whether the same genotypes do the same thing in the environments or they do different things. And what we look for is a breakdown of the correlation between genes and environment. So again, for those of you that haven't thought about this for quite a few years, what we're looking for is here at the bottom, we have, say, two environments, say temperature 1 and temperature 2. These are all the genotypes in our experiment. We're looking at some measure of performance, in this case, let's say aerobic scope. And we want to know, do they all do the same thing? So do they all decline in the same way? Or maybe there are some groups, some family lines, some genotypes that actually do quite well in the alternate environment while others don't. So this would be a high correlation. So something like this, at 0.93, we're seeing everything's doing the same thing. So there's no G by E. There's a strong correlation. But when that breaks down, and we actually see the correlation breakdown with these, which are the ones that I've highlighted in yellow here, what it tells us is that different genotypes are doing different things. And therefore, what we're thinking about here is that there are some family lines that do better than others in the altered conditions and might therefore be able to, these will be the ones that might respond to selection and adapt through, and we'll see adaptation through time. So, we've got some pretty good news, I think, in general. There's some plasticity that might be beneficial in buffering the population. Especially if we think about the rate of warming and what's actually happening, then it's possible that might be able to persist and that adaptation might even be able to catch up over the longer term. Now, we've a long way to go to actually sort of demonstrate that, but there's some positive news in terms of warming for the fish that we've been looking at. What I want to do now is tell you a different story about adaptation and plasticity, though, and in this case, using an example from some of the ocean acidification work that we've been doing. So, still thinking about those two sides of the story, but it's a pretty different outcome. And so, one of the things we found with high CO2 is that it can affect the way that fish respond to important sensory stimuli in the water, and particularly things like chemical alarm cues. Now, chemical alarm cues are just molecules, compounds that are released from the skin of a fish if it's damaged by a predator. So if it's caught by a predator, its skin's punctured and these chemicals come out, and that other fish see those as a really reliable threat that a predator's around. And it's an honest signal. And so what we see under present day conditions is that if a fish is grabbed by a predator, these chemicals come out, alarm cues, and the conspecifics will move away, reduce their activity, go to shelter when they smell those because it's a good signal there's a predator on the prowl. But when we reared the fish up under high CO2, what we found was that response to alarm cue was altered and they didn't respond appropriately to it and therefore might be subject to higher mortality. Now, if we looked at how we normally present that sort of data and what we see, this is some data from one of Megan Welsh's papers a few years ago, and she looked at the response of the spiny damselfish to alarm cue when they'd either been in control conditions under moderate CO2 or a much higher CO2 level. And these are the means and the standard errors, the way we normally present this. It's the time spent in the queue. So this is in a two-channel flume, where one side has the queue presented, has water with alarm cue, one side has just water, and it's the percent of time they spend on the side with the alarm cue. And you can see that for the fish, in control conditions, current conditions, they don't like it, they avoid it, it's only about 10% of the time they spend in that water stream. If they've been under moderate CO2, it's about 50-50. They don't show a strong response one way or the other, but they're certainly not avoiding it. And if you go to very high CO2, they start to become attractive. So that's the way we normally sort of present this data and talk about it. But if you were to dissect out that green bar, look at that, there's 425 fish tested there. No wonder the standard errors are quite low. And so if you were to dissect out that data, this isn't the real data, it's just an illustrative. What you'd probably see is most of it clustering somewhere around the mean, but there'd be a whole range of variance in those responses there. And what that means is that we've got some of either ends, for example, that might be highly sensitive or highly tolerant if we think about the phenotype. So these ones, let's say these ones up here, they're responding much more than you'd expect on average. These ones much less than average. These ones are really responding like control fish and therefore might be tolerant. Now, if this has a genetic basis, it's heritable, then again maybe the population could adapt. And so that's the question we had for these fish. we really wanted to know whether they could adapt to this problem. And there's another way that you can estimate heritability, apart from these sort of pedigrees, is you can actually just look at the similarity between parents and their offspring. And it's something we all know. We all know that kids tend to look, offspring tend to look more similar to their parents. You can see familiar relationships in people. And so we know this, it's commonly used, and we can actually what we can actually do is that we can measure the parent phenotype, we can measure the offspring phenotype, we can make a regression of that, and from that we can calculate heritability of a trait. And the particular one that we really want to look at is the father-offspring regression, because that's where we expect to see the greatest amount of genetic variance expressed, because there's less chance for maternal effects, the way the mother might provision the eggs or things like that. So the father-offspring regression is the one we're really interested in. And it turns out our little damselfish is a great model for this again. And the reason being is because it's pretty unique as a reef fish in that it forms monogamous pairs and offspring stay with the parents for several months. So it's one of the very few reef fish that we have that we can actually be sure that we can go out and catch the parents and their offspring and actually do something like this, a parent-offspring regression. And that's exactly what Megan did. So this is part of her PhD, and she went to Lizard Island and she caught pairs of Acanthochroma, she sexed them, then she got the offspring from those fish and she tested their response to alarm cue. And so we have on these graphs now the amount of time the father spent in chemical alarm cue. So what we did was she exposed the adults to high CO2 for a few days and then tested their response to alarm cue. She got the offspring, half of them she put into control conditions, half she put into CO2, and again she tested their response to alarm cue. So she's looking at the correlation between what the father did, his time in chemical alarm cue, and what the offspring did. And it turned out that there was a good correlation. We can estimate that it's highly heritable. Now you can see here, this is the father time spent in CO2. There's some that are quite tolerant. They're down this end, even though they've been in CO2. They don't spend a lot of time in the alarm cue. Others that are more sensitive. But the main thing is that there's this strong correlation here under high CO2, under acute high CO2, illustrating that the trait's heritable. And even more interesting is that there were some fathers produced offspring that had exactly the same response as their siblings in control conditions. And so they showed really no effect of high CO2. And so these ones are really tolerant. So these fathers are producing offspring that are completely tolerant to high CO2. And these might be the ones that under high CO2 conditions you'd see selection for, and that those alleles could spread through the population. So Meg did this in the field, which was exciting. We could do this for the first time with wild caught animals. And it's not very often that you can do that. But she also decided to do this in the lab as well. And here we tried to sort of stretch it out a little bit. And so what we tried to do here was, rather than just looking at sort of what's the average in the population, what Meg actually did was she got the adults and she paired them together if they were tolerant. So you'd have a tolerant male and tolerant female, or a sensitive male and a sensitive female, pair them together. So we hopefully get offspring that were sort of right at the extremes of sensitivity and tolerance. And you can see that here in the fact that this is the father's time in alarm cue. A lot of the plots or the data points are clustered around the ends rather than in the field. It's clustered more in the middle. But again, it turns out that it's highly heritable when you force them to do this. There are some potential problems with that in terms of estimating heritability, but in terms of broad sense heritability, that's fine in general. It means that yes, that these sorts of things would be passed on to the offspring. So that was cool. We could do it in the lab as well. But there's a but to this. Nothing's always as easy as it would seem. when Meg did this, we were talking about acute exposure to CO2, high CO2, so just after 4 to 5 days, both in the parents and in the offspring, and the trait was highly heritable. There was variation that was related to the father's variation. But Meg decided she was going to rear some for much longer, so she took some of the siblings that had been exposed acutely to high CO2, and she kept rearing them for several months, and then she tested them again. And after several months, there's absolutely no relationship between the offspring's time in alarm queue and the father's time in alarm queue. That heritability has disappeared. We can no longer see it. Now, these are sibling fish, so there's still the genetic variations there. What's happening is that plasticity over that time has occurred, and that we've got non-adaptive plasticity that is actually obscuring the genetic variance and meaning that the trait is no longer heritable. So yes, there's genetic variation, it's still there, it hasn't gone anywhere, but plasticity has now changed the phenotype. And it's only changed the phenotype in those ones that were originally tolerant. Through time, they lose that tolerance through phenotypic plasticity. And what the point of that is that plasticity is not always beneficial. And in this case, we've got non-adaptive plasticity. So while we like to think of plasticity as being this wonderful thing that will always benefit a population, it doesn't always, and we need to be careful not to fall into that trap. And so that's just two examples, I think, of opposite ways, outcomes for populations in response to, or the same species, in response to environmental stresses. We've also been able to use the molecular approach here as well, and this has actually been really exciting, I think. We took those same fish from Meg's experiment, so they've been reared up, these were the pairs, tolerant pairs or sensitive pairs, and we had their offspring. They were either in control or high CO2 conditions. Celia Shunter took those offspring, she took out their brains and looked at the transcriptome again, so all of the gene expression in the brains of those fish. Two things you can do from that. One is look at the mechanism that might be responsible for these behavioural changes that we see, and this seems to relate to regulation of the circadian rhythm. I don't want to focus too much on that, that's just to say you can get at the mechanism. But what's really interesting is just think about what she did here. We had fish in tanks that came from different parents. The only difference between those fish in the tanks was whether they'd been reared up under high CO2 or control, and how we'd assigned their parents, whether they were tolerant or sensitive. Nothing else. We didn't do anything else. No tests. These fish weren't tested in any trials or anything like that. We just took them out of the tank and we looked at the patterns of gene expression in their brain. The only things that we knew for sure about those fish was whether they'd been reared up under high CO2 or control conditions, and how we had assigned their parents. Then Celia looked at all the transcripts and then clustered them all together, and what fell out of this was this remarkable pattern here, where just in the brain transcriptome of these little baby fish, we could see how we had assigned their parents in terms of sensitivity. So we could see whether we assigned their parents as being CO2 sensitive or CO2 tolerant under high CO2. And this, in the differences in the transcriptome were most was most extreme in high CO2 conditions, as you would expect, because that's where those genes are being expressed. But what that tells us is that just using a molecular technique, we could already identify the phenotype of the parents. And that's another really good indicator, at least. It's not definitive proof, but an indicator that, again, it must be heritable genetic variation in tolerance to high CO2, because we could see it in how they were expressing their genes. Okay, so that's all well and good. Now there's some good news, there's some not so good news about how fish might be able to adapt to those direct physical changes in the environment. But the other thing, of course, that we've got happening on coral reefs at the moment is that the habitat that they're using is changing. And corals are more sensitive to temperature in particular, and ocean acidification for that matter, than are fish. Much more sensitive. So the fish aren't just dying from a little bit of warming, the corals are. And that changes things quite a lot. And if we just think about the last couple of years, we've seen back-to-back bleaching on the Great Barrier Reef in 16 and 17. In 16, the bleaching was severe in the northern third of the reef, in 17 in the middle of the GBR. And if we think about what that's meant for mortality, if we just look at the 2016 data, we've got upwards of 50% mortality in the northern third of the reef, and that's just from the 16 bleaching. So really massive changes in the number of corals on the reef and the coral cover and the types of coral. And this will have effects on the fish populations. Now, we know from work that we've done over the years that probably only about 10% of coral reef fish around about are coral dependent. They depend on live corals for food or shelter all of their life. But despite that, when you see whole-scale changes in coral cover, you can find much greater proportions of the fish population change than just that 10%. Of course, those 10% decline. Now, if they're dependent on live coral and you take it away, it's a bit of a no-brainer that those fish respond, and they do. But it's all the other ones that respond as well. And we're talking about whole-scale, large-scale changes in coral cover here. Up to 75% of the species can decline in abundance, and at least half of them can decline by 50% or more. And we now have two good examples of that with large-scale habitat changes. The first was Jeff Jones' work up in Papua New Guinea, where there was really widespread changes to coral cover on reefs in Kimby Bay in the early 2000s after the 1998 bleaching. And he was the first one to really demonstrate just how much of a fundamental effect this could have on fish populations, with 75% of species declining and 50% declining by 50% or more. More recently, the same sort of pattern has been demonstrated on the GBR from the long-term monitoring program. This is a paper written by Alistair Chill and others. And the similarity in that's just remarkable between these two data sets. And this is for reefs that were affected by cyclones on the southern GBR. And you see basically the same thing. Most species decline in abundance. The other interesting thing here is that just nicely done in this plot, the way they've done it, is they can actually show what types of fish are responding as well. And so there's lots of damsel fishes and butterfly fishes and the smaller things that are really declining the most. Here they've got mostly larger herbivores that are increasing. So that's not surprising either. There's a whole bunch of opportunistic things that probably don't mind too much if coral goes, and are probably quite happy if there's lots of algae around for them to eat. So large herbivores tend to increase in abundance, a whole bunch of other things decrease. Some other important things here are things like coral trout and some of the emperors and large fish that people are actually concerned about for fisheries or recreational reasons. We're all in love with coral trout. They declined as well. So even though they're not coral dependent when we think about it, they do respond to changes in habitat. So there's a couple of good examples to what can happen. And of course we've got a pretty good idea why this happens as well. As we see, changing from sort of healthy coral-dominated reef to losing the live coral but still the structure, and then maybe finally getting to something like this through time. We've lost a couple of things. One is settlement habitat, and it turns out that a whole bunch of little fish like to settle in or near live coral, even if they don't use it later in life. And even things like the coral trout often like to settle around live coral. So it might not be necessary later in life, but it can be really important early in life. It's obviously live coral makes shelter for fish in order to escape predators, and so it's an important shelter, even if it's not the live part that's important, just the structure is important. And even some of the larger fish that are predators themselves actually like having places that they can hide, either to ambush prey or avoid larger predators themselves, so they need the shelter as well. So it's those combination of things that probably are causing this decline. It's something that hasn't really been thought about too much, and it's only something I started really thinking about when I was asked to give this talk at the Gordon Research Conference, was that these two things are not necessarily, the two things I've been talking about today, are not necessarily completely isolated or independent. actually population decline from coral loss will influence the ability of the fish to adapt to climate change, and potentially quite severely. And there's three reasons for that. One is that as the population declines, we'll see a loss of genetic diversity. Smaller populations have less genetic diversity, so there's less genetic variation for selection to work on in terms of responding to climate change. When we see dramatic declines, we often see selective sweeps as well. So one thing that often happens is that the genotypes that sort of are better at coping with the environmental, let's say the changed habitat, are retained and those other ones are lost. So we can see selective sweeps. So it's not just from the smaller population, there's a sweep of alleles through the population as well. So you have even less genetic diversity left. And the other thing is that we can have genetic correlations. And so if response to habitat loss is negatively correlated in a genetic sense with adaptive potential to warming, for example, the two things will trade off to actually effectively constrain the scope of response, adaptive potential to climate change. And so this loss of the population from habitat loss is actually potentially really important to think about adaptive potential of these fish in the future. Of course, fish populations can recover. There's plenty of evidence for that. This is just recovery after cyclone. This was, I think this is work that Andrew Halford's over with the reference there, but I think that's Andy's from years ago, on down in the southern GBR, swains, I think, these fish populations coming back after they got knocked down by a cyclone. Again, they declined as a result of habitat loss and then came back. The problem, of course, we've got is that these populations, the coral just hasn't got time to recover at the moment. It keeps getting hit by a disturbance, and so we've sort of got this slide here into to declining, degrading habitats. And so the problem is that while fish might be able to adapt to warming, they can't really adapt to habitat loss. And so this is going to be a really critical thing for them. Now, while of course these days most of the work on, I actually put the slides in the wrong order here from what I wanted to say, but I'll go back and I'll come onto this and I'll go back. So, okay, they can't adapt to habitat loss. Just winding up now, some really important future directions then I think are understanding the interaction between plasticity and genetic adaptation. How does it really work? I've shown you two examples. One where it's potentially they work beneficially together, one where they don't. We really need well-designed experiments to understand the benefits and constraints of plasticity. And there's a really nice review, again, led by Jenny, who's really leading this field now, that's just come out on transgenerational plasticity and the sorts of experiments that we need to do. So if you're interested, have a look at that. And I think one thing we haven't thought about is how habitat loss might actually constrain adaptive potential to climate change, and something we need to think a little bit more about. And I'd argue that habitat loss is really the primary contemporary threat to reef fish populations in the current day. And if we don't deal with that, all we're going to be thinking about in terms of adaptation to climate change is for sort of a ragtag bunch of gobies and parrotfish and a few surgeonfishes that love living on degraded reefs, and how they're responding to climate change. Maybe it's not that bad. But you get the point that we need to think about this because not all species are going to do well in degrading habitat. And I'll leave it there. Thanks to a whole bunch of people, and particularly these people along the bottom who've really been sort of instrumental in the work that I presented here today. Thanks very much.